Hey guys, this is Mrs. Allgood. I am here with you today to talk about transpiration. Okay, and so if you remember back to fall semester, we talked about C3, C4, and CAM plants, and we talked about how with C3, which is most of your normal plants, sometimes they close their stomata to prevent transpiration. We talked about it in the water cycle too, to prevent water vapor from wanting to leave the leaves through the stomata. And so what we're mainly talking about today's notes are pretty short. This is just about factors affecting the rate of transpiration, what causes water to move up the stem of the plant, and what makes guard cells open and close. So normally we would have had a transpiration lab to do. Given the new format of the AP exam this year, I think it's something that we could talk about, as we get a little closer to the exam, but it's not something I'm worried about us doing right now. So remember that your guard cells are called that because they guard this opening. Okay, and that opening is the stoma. If we're talking about many of those openings, we're talking about stomata. Okay, so when water enters the guard cells, so when you see that, you should think, okay, well, why would water want to enter the guard cells? Well, if there's more solute in the guard cells than there is outside, then water's going to want to go in. And so that's going to make the guard cells become swollen or turgid. That just means they're filled with water. And the guard cells are joined to each other at the tips. So because of that, when they swell with water, it kind of makes them buckle in the middle, which leaves that opening, which is your stoma or stomata. Okay, so the turgidity here makes the guard cells buckle in the middle and thus open the stoma. Okay, when guard cells lose water, so maybe there's more, dissolved outside, more solute outside the guard cells than there is in, maybe that causes water to leave. It's going to lose what we call turgor pressure, which means kind of like the water pressure inside the cell. And that's going to cause the guard cells to, the stomata to close like you see here. Okay. So in general, stomata tend to be open during the day to allow CO2 to come in so we can do photosynthesis, and they tend to be closed at night. Okay. The opposite is going to be true for your CAM plants. All right. Let's talk about this. So, a couple weeks ago, we talked about the pathway that water will take as it moves through a root on its way to the xylem. So, kind of what we're looking at here is how that water moves up the xylem to go from the roots to the shoots. Okay? And so remember in your maple syrup question that you did, I asked you if maple syrup was made from xylem sap or phloem sap, and it is indeed made from xylem sap. And xylem sap is just your mixture of water and minerals that gets pulled up through the sapwood. Remember, the sapwood can still conduct water, the heartwood cannot. The heartwood is clogged with resin. Okay, we say that with water, it is more of a pulling force. Okay, think about like a pulley, like you are pulling water up that stem rather than it being pushed from the bottom. Okay, so the pulling force is transpiration. Water that escapes through the stomata, through transpiration, has got to be replaced. So that causes more water to be taken in by the roots, transported upwards, and then eventually out to the leaf. So why does water want to leave? Well, you can think of it as a lower concentration of water outside the leaf. So maybe if it's really hot, maybe if it's really dry outside the leaf, there's going to be less water out there, so water's going to want to leave. But you know what we're going to bring up? We're going to bring up water potential. Okay? Water potential is the potential for water to want to leave a given area and go somewhere else. Okay? So, if there's a lot of water vapor inside the leaf, okay, and there's not much water vapor outside the leaf, then inside the leaf is going to have a higher water potential, and outside the leaf is going to have a lower water potential. And remember, water always moves from an area of higher water potential to lower water potential. And so therefore, water is going to want to leave through the stomata. Okay, so let's look at some other forces that help pull the water up the xylem. And we talked about this way back in the very first unit fall semester. Cohesion. So what you're seeing here is you are seeing hydrogen bonding occurring between different water molecules. That would be cohesion. Makes little strings of water molecules. Adhesion, the water adheres to the cellulose in the walls of the xylem cells. So not when the water molecules are attracted to each other. That's cohesion. Excuse me. But when they are attracted to the cellulose molecules in the walls of the xylem, that's considered adhesion. Okay, and then capillary action, that is the rise of liquids up a narrow tube, like when you get your blood drawn. So those three things together are really the driving forces that help pull the water up the xylem. We already said that transpiration was a driving force. Okay, as that water transpires out the stomata of the leaf, that water's got to be replaced. So that causes more water to be pulled in through the roots. And cohesion and tension, you can kind of think of it like surface tension. This transpiration, cohesion, tension theory is designed to explain what forces are at work to pull the water up the xylem through the stem of the plant. All right, factors affecting transpiration. So this is what we would be investigating in lab. We would have little plants from Lowe's, and we'd be exposing them to different conditions, and I can tell you how we would do that. It's really not that big of a deal. And all we do for this, we actually take a plant with its soil, with its root structure, and we put it in a Ziploc baggie, and we tie the neck of the Ziploc baggie off around the stem. Okay, so we're trying to make it so even if water did evaporate from the soil, it's still going to be stuck in that bag. Okay, and we basically mass the plants on day 1 and day 2. We mass the initial mass of the plants, and then we expose them to conditions, whether it's light, whether it's heat, whether it's humidity. We expose them to that for about 24 hours, and then we take their mass a second day. So first up is temperature. If you have a higher temp, at least up to a certain point, you're going to have a higher rate of transpiration. Okay, why? Remember, at higher temps, particles will have more kinetic energy, and so that is going to cause evaporation to happen at a faster rate. Okay, meaning that if you have a leaf, okay, let's draw a little leaf here. Okay, if you have this leaf, okay, and you have maybe some little water droplets, okay, in the air outside that leaf, all right, if it's hotter outside, that's going to increase the rate of this evaporation, which will tend to keep the water potential fairly low out here outside the leaf, which is going to cause water from inside the leaf to want to leave. That's a lot of leaves and leaves. Anyway, up to a certain point. If we make it too hot, the plant can actually get kind of stressed out. Like sometimes you guys might get stressed out from time to time. And it can trigger stress hormones like ethylene. Okay, possibly even abscisic acid, but probably mainly ethylene can be produced. So when we do this, what do I do? With temperature, we actually put it in the incubator, like where you put your little bacteria plates. When we were making them glow in the dark in December, we actually put it in the incubator at about 37 degrees Celsius, which is not going to fry the plant. It's not going to kill it, but that's a pretty good temperature to test it at. All right, humidity. Hopefully this makes sense. Higher the humidity, meaning there's more water vapor in the air outside the plant, the slower the rate of transpiration. Okay, so again, if we have this leaf, okay, there's our leaf, And if we have all this moisture out here, moisture, I don't want to draw that many raindrops, okay? But let's say this is all water vapor in the air, and I've only got a little bit of water vapor in there, okay? Remember, high water concentration is high water potential. So this is going to have a low, I was trying to write the word low. I don't know if that's going to happen. And there's a low water potential in the leaf, and there is a high water potential outside the leaf. Okay? Water doesn't move from low water potential to high. It's not going to want to leave if there's already a lot of water vapor outside that leaf. Okay? So how we do this, we typically just get like a gallon-sized Ziploc bag and we spray the inside of it with water, and then we just put it over the plant. We try to create like a humid atmosphere for the plant, and you shouldn't see much transpiration going on in that plant and lab. So remember that water potential is a measure of the potential of water to want to leave one place to go to another. And so if there's a lot of water vapor outside the leaf, kind of like I was trying to show you with the picture here, water inside the leaf will not want to leave as much, okay? So if there's a lot of water in here and not much water out there, water is going to want to leave. But if there's a lot of water out here, there's no way that water is going to want to leave there. So as the relative humidity increases, okay, as we get more and more and more humid, the transpiration rate should go down. Okay? Air currents. So for this one, again, we're not trying to activate any stress hormones here, but typically we just sit our little plants in front of a little tabletop fan. Okay, about a foot away from the tabletop fan. So strong air currents or wind should lead to higher rates of transpiration. And the reason for that is that wind is blowing any water vapor that's there. It's going to blow it away from the leaf, making the area just outside the leaf, just outside the stomata, it's going to make that area fairly dry. And so because of that, it'll have a low water potential outside the leaf, a high water potential inside the leaf. Water always wants to go from high to low, therefore it will leave the leaf. Okay, so wind moves the water vapor away from the leaf, resulting in decreased water potential outside the leaf. This is just your solute potential equation. We'll come back to that if we need to practice that before the AP exam. So as the wind velocity, like wind speed, increases, the rate of transpiration should also increase. Okay, light. This is one that we typically use our floodlights for, but I usually put a beaker of water, like a big beaker, like 1,000 milliliter beaker of water, in between the floodlight and the plant. Because if I don't, the floodlight gets so hot that it will activate stress hormones in the plant, and we're trying not to measure heat here. We're trying to just measure light. So the water in the beaker will absorb the heat energy and just pass the light through. So the technical reasoning for this is that light stimulates guard cells to take up K+. That'll give the guard cells more solute than the surrounding solution. That will make water want to enter them and make them turgid, which cause stomata to open. That's more complicated. To me, the more easy definition or explanation here is that if you're going to have more transpiration the more light you have, and that's because during the day, stomata are going to be open to allow CO2 to enter, and if those stomata are open, the CO2 can still exit. So notice here, you have light intensity and rate of transpiration. So up to a certain point, as light intensity increases, the rate of transpiration will also increase. But notice there's a certain light intensity that we get to where it's not going to raise the rate of transpiration. There's simply like nothing more we can do here. You've maxed out your max light intensity. Okay. Evolutionary adaptations to control transpiration. So cam plants, remember, only open their stomata at night. This is another part of the transpiration lab that I like doing, where we kind of imprint the stomata from a leaf. The stomata are located typically on the underside of a leaf. If they were on the top side, they'd be directly exposed to sunlight, and you'd probably be losing a whole lot more water through your leaves. And then in gymnosperms, like conifers, the stomata are located in deep pits, which is going to also help reduce their rate of excessive transpiration. Okay. And then finally, there's this topic of guttation. Okay, it says guttation is the appearance of drops of xylem sap on the tips of edges of leaves in some vascular plants, such as grasses. The way you sort of want to think about this is that water got pulled up the xylem because the stomata were open. Remember, it's a pulling force that's pulling that water up the stem. And then the stomata sort of closed all of a sudden, and this was the little droplet of water that was left. We don't want to confuse this with dew. Dew condenses from the atmosphere onto the plant surface, but it will also condense onto other things like your patio furniture. Okay, if it's guttation, it's really just on the plants and nowhere else. I think that's it for today. I tried to make the notes a little bit shorter. Tomorrow shouldn't be too bad either. There's really no review sheet for this today. Normally our review is our lab. So just make sure that you get the plant walk Google Forms quiz done. That's due Tuesday night, I think. And then if for some reason you did not do the secondary growth and stems quiz before spring break, please go ahead and do that also. Thanks. I'll see you guys or talk to you guys tomorrow.